Yeah, good morning. Today we are going to learn protein, structure of proteins, and even types of amino acids, and what are the different approaches the proteins involved in our daily life. Okay. So what are proteins? Means proteins are the most abundant organic molecules which are present in our body, and these proteins are made up of different amino acids. As we know, in daily diet we require carbohydrates, fats, and even protein. So previous classes we gone through the different types of carbohydrates and their significance and their classification. Even in terms of fats we have learned, right? Today in this classroom we are going to learn about proteins. So proteins commonly known as building blocks, right? We commonly known as building blocks. So these proteins are involved in our daily cell metabolism. As such, carbohydrates, how they involve, then even fats, how they involve. In the same way, the proteins can also involve in the building block, right? From cell metabolism. So they are also commonly known as bricks and the mortar of body. So what type of functional groups are present in the proteins? Means we have the two different functional groups, namely carboxylic group and amine group, right? So this carboxylic group and amine groups are those two functional groups which are responsible for the formation of different groups of amino acids. So in a classification of amino acids concerned, we have the three different types of classification. One is acidic amino acid, basic amino acid, and neutral amino acid. So among these two, carboxylic acids, right? COOH, either they are acidic group or basic group. Acidic in nature, right, brother? So if more number of carboxylic groups will be there, they are considered as amino acid is acidic in nature. If more basic amine groups will be there, such type of amino acids are known as basic amino acid. If those amino acids having equal proportion, what we call them as neutral. Are you getting me? Clear? Say yes or no?
Yes,
right now. So carboxylic groups are there and amine groups are there. Right now we will go with one how these amino acids are going to be formed a linear chain. So when you see across after the cell, in this cell metabolism, after the transcription and translation, at lastly we will get the polypeptide chain, and this polypeptide chain is a linear chain. Clear? Such linear chains cannot be involved in direct in cell metabolic function. So they should undergo some changes. They should undergo some changes. So what type of chain? The linear chain of polypeptides is going to be converted into folding, primary folding, secondary folding, and tertiary folding. So with the help of the molecular chaperones, here what we call molecular chaperones are those proteins. They are responsible to convert the linear polypeptides into primary, secondary, and tertiary structures. Among them, we have secondary proteins, tertiary proteins, and quaternary proteins. So before we go for the discussion, now again we want to know this is the first amino acid and second amino acid. How these amino acids are interlinking between one amino acid to the another amino acid. That is the major task in cell metabolism, right? So how many functional groups were there? In an amino acid, how many functional groups are there? Two, namely carboxylic group and amine group. So this is the first amino acid. Amine group, carboxylic group. So this is a red color here to here is a first amino acid, and this is a second amino acid. So first amino acid and second amino acids are interlinking. How? Carboxylic group of first amino acid and amine group of second amino acid, they are joined collectively and they remove the water and they form a strongest bond, what we call peptide. So the strongest bond, the peptide bond is formed between carboxylic group of first amino acid, amine group of second amino acid. Eliminate the water molecule to form peptide. So this is the peptide. Are you getting me?
Yes.
So such type of peptide bonds are formed. Here first peptide, second, third, fourth, fifth, third, right? So going on. For example, here how many amino acids are there? 1, 2, 3, 4, 5, 6, 7 and 8. So how many peptides will be formed? How many peptide means? N minus 1. Here we have N minus 1. How many N? 8 minus 1. How many? 7. Count here. 1, 2, 3, 4, 5, 6 and 7. So when you are preparing any entrance examination, this is the one task where they will going to get MCQs. Okay? Now, so apart from the 16 amino acid, how many polypeptides will be there? 15. Nice. So here, so after the completion of the formation of peptide 1, now we will go with the structure of proteins, right? So just now I explained about the linear polypeptides which are there in a cell metabolism. They are unable to do cell function, so they should undergo some modification. So we call them as a primary, secondary and quaternary foldings among them. So linear proteins are not involved in active metabolism when compared to secondary, tertiary and quaternary. In case of secondary, proteins are classified into two different types, namely alpha sheeted helix and beta sheeted helix, right? Alpha sheeted. So it looks like in a chain-like process which is having the helical in nature, where it is most commonly found in all proteins. Most of proteins, they are in alpha helix. Now, they are tightly packed in presence of hydrogen bond. Extensively hydrogen bonds will be there. Are you getting me?
Yes, sir.
When compared to linear proteins, secondary proteins are active. When compared to secondary protein, tertiary and quaternary proteins are more active. Are you getting me clear? Right now. So this is a one case where we found an alpha helix, and the beta helix. The scientists by the name Pauling and Corey was the scientist. These two scientists studied on beta sheeted. Beta sheeted means, you know, one layer is covered with another layer, one layer is another layer, it is covered with another layer like this. Are you getting?
Yes, sir.
So what you call sheet-like structures, right? So in the same way, beta sheeted, this when you think this two red color is an alpha sheeted and this one is a beta sheet, that means they are overlapping between the one sheet with the another sheet. In the same way, example, when you found in insulin, insulin is having the two peptides. One is alpha peptide, second one is a beta peptide. So alpha peptide is, this is an alpha peptide is having 21 amino acid, and beta peptide it is having 30 amino acid. Alpha polypeptide, beta polypeptide. So these two linear polypeptides are interlinked between disulfide bonds, intracellularly or extracellularly. They are made up of disulfide bond. For example, this is a alpha sheeted and this is a beta sheeted. If you open like this, it will open. So when they form a disulfide bond between these two structures, to pull it is too tough to be separate, right? So these polypeptides in artificial approaches, when you are learning in your second year or maybe final year of your BSc, go through that the structure, how they genetically engineered to construct an insulin. We have the topic. We will discuss in that topic also, right? Now, so this is about beta sheeted structure. Now tertiary. In the tertiary structure is a three-dimensional protein structure where it is having sulfur bonds, disulfur bonds, ionic bonds, hydrophobic bonds, hydrophilic bonds. Where we found among them? We found such a polypeptides of different amino acids. Why I mentioned the different types of bonds will be formed, because 200 amino acids are not similar types. There may be aliphatic amino acid, there may be sulfur containing amino acid, there may be hydroxylic group containing amino acid, amine containing amino acids. Okay, are you getting the concept clear right now? So different amino acids to form the tertiary structure, there may be a bonding pattern will be differ, the dimensional structure of the protein will be differ. So here we have a hydrophobic bond formation, hydrophilic, ionic, etc., etc. Clear? Then quaternary structure. So this is a quaternary is highly coiled one. Hemoglobin, myoglobin, and even lactic dehydrogenase enzymes. All these enzymes, you know, myoglobin will carry oxygen for different body parts, whereas hemoglobin will carry the oxygen, whereas myoglobin will carry the oxygen for muscle tissue, right? Lactic acid dehydrogenase. All these enzymes are tightly coiled when compared to tertiary. Here we have tightly bondages of polypeptides. Are you getting the concept? So even they are in a metabolic function, they will carry the different products, namely oxygen they will supply for the various body parts by hemoglobin, and oxygen can be supplied to the tissue, muscle tissue by myoglobin, etc. Are you getting? So all these things are the structures of quaternary proteins. Quaternary proteins go through the quaternary protein example, tertiary protein examples, then even beta sheeted example, alpha sheeted examples, and even how many types of bond will be formed between the different types of proteins which are going to be learned, right? Now, apart from that, now we will go with the study of the different amino acids. Amino acid. Amino acids are a sub-monomeric compound of a functional group. So these amino acids are classified into three different types. Just now we have discussed. One is if it is having amine group, if it is having carboxylic group, or if it is having equal amounts of amine and carboxylic group, they are classified into amino acid, namely acidic amino acid, basic amino acid, and neutral amino acid. Acidic amino acid in the sense more acidic group will be there. Basic amino acid in the sense more basic group will be there. Neutral means equal amount. That means jitter ionic compound. So when you see the glycine is having aminic group 1, carboxylic group 1, right? So here aliphatic, amino acids are classified into various types. Among them also we are studying aliphatic side chains. So one side chain, two side chains, three more different side chains are there, right? Are you getting? So here glycine, alanine, valine, leucine, and isoleucine, these are all an aliphatic amino acid. So I don't have the time to draw all these things, so I shortcutly mention to by heart all these things when you are preparing for any entrance examination. Sulfur containing amino acid, that is cysteine methionine. So previously in Gurukulam also when we wrote an exam, we found sulfur containing among the following, which one sulfur containing amino acid? That one is cysteine methionine. Amino acid with amine group. So these aspartic acid, aspargine, commonly we call them. Glutamic acid, glutamine. These two are amide containing amino acids. And here proline. This proline is highly important when we study about stress tolerance. You know, plants will stress tolerance during the hot summer. Even plants can tolerate hot, wind which blow fastly. In seashores, sea outside shores, what they do, they planted subabul plants mostly, right? When blow will ooze from one side to another side, it will bend like this. We think that the tree will be break down, but no. After some time, again it will retreat back. That means who is going to be overcome by the stress condition means which amino acid? Proline, right? Even plants which grow in salt conditions, even the plants which grow in drought conditions, even the plant which are in high salinity in nature, the plants can withstand for that environment. The remaining plant will be died. The plants which withstand, which is having more amount of proline. Are you getting the concept here? So this is about proline containing amino acids. But that means proline is a stress tolerance where it is having pyrroline compound among them. So here basic amino acid, lysine, arginine, histidine. These are all basic amino acid. Important beta for this one. Aminosole containing compound containing amino acid is histidine. Eta amino acid containing compound is lysine. Don't forget, all the compounds are important. These are all aromatic amino acid. Aromatic means smell releasing compounds, right? So here we have phenylalanine, tyrosine, and tryptophan. Indole containing amino acid is tryptophan. Don't forget that one. Okay. All are important beta. Now, hydroxylic group containing amino acids are serine, threonine, and triosine. So these are all few amino acid structures where I wrote, and even I wrote different types of functional groups which are present in the different amino acids. These are all important for your any entrance examinations, and even you are preparing for any competitive board examination point of view. These are also important, right? Thank you.